Hello student, this is Miss D.A. Marvekar, working as Assistant Professor in the Department of Zoology, Rajashri Chhatrapati Shahu College, Kolhapur. In our previous lecture, we have already discussed about the glycolysis, in which glucose was broken down into the 2-pyruvate molecule. Now, what happens with respect to that pyruvate? Let us begin one new topic, that is Kreb cycle, or it is also called as a TCA cycle. Now, if we are considering the organisms, we will divide that organism into two parts. One group is aerobic one and another is anaerobes. In case of anaerobes, they will not require the oxygen for their metabolism. But in case of a aerobic group, they will require the oxygen. So therefore, if you are considering the aerobic organisms, in that aerobic organisms, whatever pyruvate is generated in the glycolysis, that will get oxidized with the help of this Krebs cycle in the mitochondria. So most of the important metabolic pathways which are related with this energy production in the animal system. So all enzymes which are catalyzing all the reactions which are belonging to the Krebs cycle, which are located into the mitochondria. So the location of the mitochondria is the location of the TCA cycle or the Krebs cycle is the mitochondrial matrix. As the location of a glycolysis is the cytosol, after glycolysis, the pyruvate which is formed, it will enter into the mitochondrial matrix so that it will proceed in the form of a Krebs cycle to generate the energy. Now, the question arises how the pyruvate which is present in the cytosol will get entered into the mitochondrial matrix. So, with the help of a pyruvate dehydrogenase enzyme, which will convert this pyruvic acid into the acetyl CoA, and due to this enzyme, this acetyl CoA which is formed, it will get transferred from this mitochondrial membrane boundary to the mitochondrial matrix. Now, how this process takes place? So, this pyruvate dehydrogenase enzyme, it will require the 5 different coenzymes and it is a multi-enzyme complex. That is, multi-enzyme complex means the enzyme which is having a more than 2 enzymes which are located nearby each other, and it will form a major enzyme. That is, it consists of the number of enzymes which are present at near together so that the reaction will get speed up. So this is also the multi-enzyme complex which consists of the 3 different enzymes. We will refer that enzyme as a E1, E2, E3. E1 enzyme is nothing but pyruvate dehydrogenase enzyme. E2 enzyme is dihydrolipoyl transacetylase. E3 enzyme is dihydrolipoyl dehydrogenase. And apart from these 3 enzymes, that pyruvate dehydrogenase complex required another 5 coenzymes which will speed up this reaction. The one of the coenzyme is TPP, that is thiamine pyrophosphate. Another coenzyme is flavin adenine dinucleotide. Then coenzyme A, then NAD, that is nicotinamide adenine dinucleotide, and the last one, that is the fifth one, is a lipoate. So with the help of these 5 coenzymes, these 3 enzymes will catalyze and convert the pyruvate into the acetyl CoA. Now we will study in detail about how this process will take place in this simple manner. Now this arrow, what this arrow is indicating? This arrow is indicating that pyruvate will be converted into the acetaldehyde TPP and it will release the CO2. Okay. So the pyruvate dehydrogenase enzyme belonging to the multi-enzyme complex, it will first convert that pyruvate, it will allow to react that pyruvate with the thiamine pyrophosphate enzyme, pyrophosphate coenzyme, and it will remove CO2, that is decarboxylation reaction takes place, and here this TPP will get attached to the pyruvate and it will form this acetaldehyde TPP. Now, this acetaldehyde TPP will react with the oxidized lipoate which is formed in this cycle, and this oxidized TPP will get converted into the acetyl lipoate. That is, acetaldehyde TPP will give its acetyl group to the lipoate. So the TPP will regenerated. It will allow to recycle here, and the acetyl lipoate moves further. So now with the help of enzyme dihydroxylipoyl transacetylase, here the acetyl lipoate, from the acetyl lipoate, the acetyl CoA will get removed, and this lipoate, this reduced lipoate will again converted into the oxidized form. During this reduction to oxidation conversion, the FAD molecule is reduced into the FADH2, and this FADH2 is again reduced this NAD into the NADH. So the net reaction consists of the pyruvate molecule is converted into the acetyl CoA by removing one carbon dioxide and by formation of a one NADH. So in the glycolysis there are two pyruvate. So that two pyruvate will be catalyzed with the help of this enzyme, that is pyruvate dehydrogenase enzyme complex, and by passing this reaction, this typical reaction, the two NADH are formed and that pyruvate is converted into the acetyl CoA. And now that acetyl CoA will enter into the mitochondrial matrix via this boundary, and then that acetyl CoA is now ready to enter into the TCA cycle. So that's it for the today. In the next lecture, we will discuss in detail about the TCA cycle. Thank you.